every time I started like digging into some of the data I was going to present, I realized that there is a lot of difference in the way that data is reported, in the methods that are used, in some of the changes that are trying to be made for the future that I thought would be useful to go over. So that's what a lot of this is. It's putting in context what's testing's being done and how it's being done. So a little bit of history. The Beaches Act of 2000 basically required states to put some testing in these parameters in place. But the parameters, the thresholds, really go back to the 1980s. And that may seem like very old values to use, but the testing and the correlation studies that were done then really seem to hold up in many ways. So, there's the definition of what a shore is, what a beach is. I'm going to show also some of the thresholds depending on the usage of an area as a beach or for other recreation, and why we still really stick to one threshold for mostly for clarity reasons. Okay. So what's being tested? Total coliforms, which is at the bottom here. I'm going to not talk much about that. That is more of a very broad term that can refer to a number of types of organisms that could potentially be due to fecal contamination, but sometimes not. As in many cases, the technology improved, then tests moved to things like E. coli. E. coli are indicators of fecal contamination. Um, in both cases with E. coli and Enterococcus species, we're looking at contamination from warm-blooded animals, and a number of birds, as we mentioned, geese, big problem, ducks. Um, but E. coli is mainly used for freshwater. Most of the testing we do is really in brackish water. So Enterococcus species have been found to be a better indicator in those types of waters, in that type of water. So that's the testing we do. That's why it's in bold, tests for Enterococcus. So when we look at the values and the threshold, so this reference looks relatively old because it is relatively old. What we consider for acceptable levels for healthy swimming, uh, for safe swimming, is this less than or equal to this circled number of 104 colony forming units per 100 milliliters. This is a very direct count. I'm going to talk about the different methods that can be used in a second, what we used. And it's all correlation studies. They basically looked at acceptable thresholds for a certain number of illnesses, um, gastrointestinal problems, even though other problems can result from exposure to bacteria associated with fecal contamination. And in marine water, along the lines of brackish water, the cutoff for designated beach swimming areas, high use beaches is 104. So that's what's been used for decades. Now, when we look at freshwater, which Enterococcus can be used for both fresh and brackish, the cutoff is somewhat different, but it's not that much different, right? In freshwater, what I should say is not that much different when we look at the sites that we tend to test in freshwater are these non-beach areas. So for some restoration projects, when we look at some freshwater non-tidal, they're areas that you don't expect people to be submerging in, but you can have people splashing around in there a lot. For pets, this has become a much bigger concern for people's dogs and pets. The values are not significantly different. There has been discussion about, well, if there are these slightly different values, why don't we report different thresholds? Part of what we consider our job is to make it digestible and understandable for the general public. And it's difficult enough to, have these indicator levels. So we stick with the 104. And because, as I will tell anyone that will listen to me, this idea of 103 is healthy and 105 is unhealthy is really in some ways a false statement, that it's very much about things like health status, which I'll talk about in a couple of slides also. So that gets to the point of, why is some of the way that reporting done problematic? A number of um, apps or databases want to report in this sort of green means safe swimming, red means unsafe. And so this in the background is part that comes from the Swim Guide app. A lot of communities request that we report to Swim Guide. We do that. Um, there's a little bit of issue with that, though. As I mentioned, 103 being healthy would give you a green, 105 would give you a red. Not a huge significant difference. In addition, the way that values are reported can differ. So we use over, as mentioned 2 slides ago, for individual measurements, it's this 104 threshold for daily tracking. Over a season, we use geometric means, and that's for looking at chronic water quality. So that's looking seasonal. And that's what we do for some of the reporting when we do season-long analysis, when Sally reports for the Magothy River Index, which I'm going to show a figure from that to refer to that. We're looking at sort of that long-term data. In addition, the geometric mean helps to sort of, quell some of the fact that since it's living organisms and there's, this sort of logarithmic exponential scale, it kind of helps to remove a little bit to the effect of some of these extreme values. A somewhat newer value for water quality is this statistical threshold value. This measures the frequency of spikes. So this is a newer value that you see being used that is this 130 number per 100 milliliters. As you saw on 2 slides ago, I used the term CFU, colony forming units. It's basically, we think of it as the number of bacteria per 100 milliliters. Most probable number is more of an indirect count by doing some type of indirect reaction or indirect calculation where you're not directly visualizing colonies. And so some places use that, and I'll explain the method in a minute. Blue Water Baltimore uses that. But this statistical threshold value, that's based on the fact that you shouldn't have more than 10% of the values within a season over that threshold. It's still somewhat unclear to me from a number of readings where they got the 130 exactly. But to me, from the reading of it, that is more of a value over time. So the fact that some areas use it for reporting as what they consider for healthy or unhealthy for a single test can be a little problematic. And that is a difference between how we do this and Blue Water Baltimore. We also are still in line with what the Department of Health reports. The Department of Health for Anne Arundel County still uses the 104 threshold also. So that gets to sort of slightly different ways that things can be reported. And then when we look at the results, so we do at Anne Arundel Community College at the Environmental Center, we consider ourselves a way to complement what the health department can do. They only have a certain budget. They focus on the areas that truly are swimming beaches, that they expect people to be, fully submerged, hanging out on the beach, that type of thing. But a number of communities have docks, piers, where people may be swimming off of, or they're definitely having significant water contact, kayaks, boating, etc. So we complement by filling in some of those sites for communities. We test every week, regardless of whether there's been rain or not. The county and programs in Maryland by the Department of Health, they do not test right after rain. This block here with the preemptive rainfall advisory, if it's rained, they consider a significant rain event, 0.5 inches or above. They just assume the water's not good, it's not swimmable, you should stay out of it. They talk about closing beaches, but that's problematic because there's no gates in many places and there's no guard saying don't get in the water. So then they report that over the last 20 years, 98% of the time, the Maryland beaches have cleared for swimming. That's true for when they test, but they don't put in there what percentage of the time the water is assumed to be unswimmable because of rain. And why we're interested in testing rain or not rain is for several reasons. Some locations, even after it rains, the water quality looks great. Eastport Yacht Club site, right? Typically, other than last week, that will show, typically, even after it's rained, that site, the values are below the threshold. In addition, we would like to see after rain if there is a trend toward the water being more contaminated from this massive amount of runoff that's coming in. So this is why when we look at data from the Department of Health, they tend to have very few numbers that are over the threshold because they just do not go out and test after rainfall. And they have a different, somewhat different goal than we have also. So I'm not saying that what they do is wrong. Their goal is just somewhat different. So then why do we then test after the rainfall? And I've, of course, sort of already given the answer to this, but we test because we want to know how bad it is right after rainfall. So this is a map from just from data from last Thursday. Each red marker is a place where sampling was done. This is Spa Creek here, and then Back Creek. Obviously, two tributary creeks that flow into Severn River. There are also sites on the Severn River that are done through a different, that are sampled on Wednesdays, not Thursdays. This was sampled Thursday, day after a huge storm that came through, somewhere around an inch and a half, 2 inches of rain. And the numbers were awful. So without even looking at the numbers, you can see every single site, if it would be blue or green if it were below the threshold. Everyone was above the threshold, even Eastport Yacht Club. So why we want to test is after rain is because this tells us that there is something even more significant about this testing date, right? Eastport Yacht Club usually is great all the time. We tend to have some of these sites that are midstream in Spa Creek and even toward the mouth in Back Creek that are usually good even after rain. So what does this mean long term? I will say, as Virginia mentioned, every time we have a day sort of like this. I will say this is the first time not a single site was acceptable, was below the threshold. But every time we have a day where the counts are very high, the concentrations are high, it makes me truly sad to have to report to people that that's what's happening. But usually there are sites that are also good. So this is us like looking at putting it in long-term trends. I'll mention some of that toward the end also. So we want to know this information. And then it also brings up the, well, what does it mean, right? What does, if we look a little bit at the counts, we have counts all the way up over 20,000, right? That water is seriously, seriously contaminated. Even at, when we look at counts, even Eastport Yacht Club at 298, which is, almost 3 times the level, what is the risk in some ways at those two comparable sites? Well, what we know is that the higher the concentration, the higher the correlation, the higher the risk of illness. And the most studied, the correlations were really mostly done with gastrointestinal illness, where you can have problems with, you can have ear infections, you can have skin infections. If you have pets, you can have multiple types of infections. But for one thing, it's the being below the threshold is not a guarantee of safety, and being significantly above it is not a guarantee that you're going to be ill. So another reason that we want to test after rain is because if you live near the water or you go areas where they're near water, there are things like sailing clubs. My kids always belong to sailing clubs. No one told them to stay out of the water after it rained the night before. No one told them to do that. They're not going to stay out. So to have that context of how bad it is and have like maybe some parents say, well, at least you want to make sure that you shower really well after you're in the water. So that's what we're working toward. And we've also found that health status is often the most important factor. Those that are really young, those that are very elderly, open cuts, immunosuppression, or if you're immunocompromised, that can have a significant effect on your health risk. So this is why this idea of just red versus green is really not truly informative enough, that it really is reliant on a person's health status and own risk. Another thing that we always focus on or make sure that we say is that, we test for one genus of bacteria, Enterococcus, right? That test doesn't test for other possible pathogens like Vibrio species and some of the Mycobacterium species and other genera that can cause infections. In some cases, there just is not good correlation between concentrations and illness. They are often species that are naturally occurring in the water, and so you just expect to find them there. There's been some studies by things like by NOAA, for example, to do some correlation between temperature, salinity, likelihood of higher concentrations, and infection, but the data's not good enough to really have these sort of thresholds. So then the question might be then why do we test it all, right? If it's, we can't say that you're for sure going to be sick or you're for sure going to be healthy, depending on the values. Why do we test? Well, one is the fact that you, if you're informed about what's going on, you can make a decision based on how bad it is, how much rain there's been, knowing the history of your location. And we also test because we find important things out. So this was from 2023. I'm not going to show huge tables of data mostly, but it was a non-rain day. And yes, up in the headwaters at some of these sites, we see CG and SEM, the values are often somewhat high. But on this date, none of the areas that tend to sometimes be high were high. Things were low. But at this one site, it was almost 20,000, which is a huge number. So usually after rain, when people want to say, "Oh, there's been an overflow of the sewer," usually we're like, "No, it's the runoff." This day, no rain. There was something going on. So one of the Spot Creek, Donna Jefferson with Spot Creek Conservancy, she reached out to the city, to Annapolis, and yes, they found that there was a blockage and there was a backup, and yes, there was actual sewage making its way into the creek. So this was kind of discovered because of the first notice that these values are really high. So it was a way to find something important out and then be able to tell the public about it, and to be able to then also compare to after rain, values are high all over, but when something like this happens very locally, you get this very local effect. Also, one other example, in Herald Harbor over on Severn River. I know this looks like a lot of data, but there's some down here around the block. Herald Harbor, this shows where their swimming beach is, and we test right off this beach. And sometimes they have issues after rain, but right early in the season, they initially, right away, we saw that there were these numbers that were high, regardless of even when there wasn't rain. Like most of the values are in the black, they're nice and low, there hadn't been rain. And they usually don't have a problem with that. So a couple things we started to know. One, these two houses that are in squares, those are newer houses. They hadn't been there a handful of years before. So more impervious surface. They kind of changed the path that we take to get to the beach. And they added a lot of wood chips. And so just basically less like areas that was a little bit less pervious, I guess is the word. And there's this one area, there's this very marshy area, and then a little outlet out where there's some a pipe, a small pipe, but an outflow. And when I discussed it, I had the tech that does the testing. We did a like just testing here and then moving as far over to the private dock over here as possible. And we saw right away that the values were immediately like very low. And what we found was that there's something, whether there was something moving from the hard surfaces from the new house or because of even animal activity, early in that season, it'd been dry and there'd been no rain. And so there was just something that was sort of just hanging, that was causing some contamination that was just kind of trickling out here and not causing extremely high numbers, but high enough to be over the threshold. After the first rain, I think that happened down over here. We basically got this column, 28 June, when you see everything else, a lot of other values are high. There was a washout effect, and it seemed to have washed out whatever was hanging out, whatever was lingering, whatever contamination that was trickling its way into the water. So it wasn't as though we found out some huge thing to solve it, but we definitely were able to at least determine a little bit of what was happening. So for awareness, if nothing else, we had told people if this happens again, if you're worried about it, you need to be swimming away from where this outlet is, where this outfall is. So there's definitely things that we've, learned from this that can be useful also on the spot. I won't do more examples because I want to move to the methods that are used. So this is a place where things are in transition, not as though it's happened yesterday. This is multiple years, but there are multiple methods that are now available. The one that we use, another one that's very popular, and then one that they keep talking about coming into use. So these are the three main methods. The EPA 1600 is microbiological. We actually see colonies. That's what you see there. We count these colonies that are selected for and inhibits other type of bacteria. The Enterococcus also can cleave a specific sugar. We produce this blue color. We can directly see what's happening. These are colony forming units because it's direct. We get the result the next day. There's always been this push to why can't we know right now, right? Why am I finding out about yesterday? So, and originally the microbiological way was a 2-day growth. The way we do it now, the EPA 1600 is 1 day. But IDEX, a company, they have a couple of methods. It's indirect. There's an enzyme activity. Again, you have a cleavage of a specific molecule and it produces fluorescence. You're not looking at colonies. That's what you see down here. There are these little wells and it kind of self-dilutes. And you basically, the next day, count how many squares are fluorescent and you go to a table that gives you the most probable number. That is by far right now the most popular method that's used. Um, it's ease of use. It's less training. You're just looking at fluorescence. You're not making judgment calls. You don't have to um, make media. You don't have to use as much aseptic technique. Um, it's also overnight. I am not a huge fan of that. I'm old school about this. You can't go back and check. You don't see the colonies. There are some issues that I worry about with salt concentration. It's not really clear how much salt inhibition there is from salt water. Um, and they basically have cornered the market on this, right? They're the only company that has this. So they could, and it's much more expensive than what we do also. The newest procedure is quantitative PCR. It's basically amplifying, looking for targets in the DNA, right, in the specific targets for the bacteria and DNA. This is also indirect. If you're looking at gene targets, you're not directly looking at living cells. You're looking for targets. It could actually just be DNA that's in the water. Again, you're looking for sequences that are specific to those bacteria. In theory, you can have it the same day. So you can have the data later in the day. It's using molecular techniques. Why is everyone not doing this now if you can get the data the same day? Well, there's good reason not everyone's doing it now. A paper a couple years ago said it's still only, at that point, it was like 1%. Like Chicago uses qPCR. I met with people from Anne Arundel Department of Health last year, and they said that they're working toward being able to use qPCR, but I have not heard if they're trying to make that happen. Why isn't everyone using qPCR? If it's the same day, great. There's a number of issues, technical and infrastructure challenges, the training for this, the detail for like pipetting and getting the procedure is a lot more complex. Infrastructure, it requires a piece of equipment that's at least $20,000 just to get the ball rolling. The consumables are also expensive. In reality, in functional time, it's not really real time. It would be a very long day. You could, but by the time you collect, you have to collect very early in the morning, get them to the lab. By the time you process, get the results, analyze and report, you'd still be talking about getting it like 8 or 9 o'clock. So for a large facility, you could do that. In a lot of facilities, you're just not going to have the ability to stretch out that day. So in functional terms, it's often the next day. As mentioned, cost constraints. It's much more expensive. They keep talking about the cost coming down, but even the consumables are still very expensive. Inertia, the cost to get this set up, it's going to take multiple years of co-reporting the way it was done and the new way to convince anyone. Public trust and communication. Uh, the values are seen very differently. There is validation literature, but everything is still reported in things on like the exponential and the log scale. They're like, oh, log 2 has this much increased risk. The public doesn't understand that. They want just a number. Just give me a number. In addition, this is showing data that you get from this. You don't look at anything directly. Things are very much based on this like 100,000 type of scale. People are used to seeing numbers that they can trust that are down into like 2, 10, 20. Numbers less than 100, less than the threshold, the confidence level is very low. So it would be it's a shift in paradigm for how people think about this. In addition, there's not as much validation as there is for the other methods. I've read several papers and they talk about, well, at this beach we've done it, but at this type of beach we haven't. So there's also the trust factor. Um, Okay, so this is why we have not even tried to convert to this. We've done some studies in the lab, and it's very hard to convert targets in DNA to known living cells. So we have tried, and we are no way confident with the results, at least that we've gotten. But the other problem, this persistent problem, is even if we know the concentration, where is it coming from? Is it human waste? Is it dog waste? Is it deer? Is it geese? Um, and this has been a challenge also. So source typing is a method that there are historic ways to do it that are not molecular, but there's really been this push to try to do this at the molecular level because the older microbiological ways were really not very easy to tell the source either. The idea here is that you're not really, in reality, you're not looking for targets that say that a human was in the water. You're looking for targets for bacteria that are in a human's feces. So it's like 3 steps back. So that has made it very complicated. So in theory, source typing could determine the source of the contamination. It provides a correlation between species-specific targets and that have been tested for known species and what's in the water. It also doesn't provide a concentration for what to know what the living number is. It's just the targets for these specific species. Um, and the companies that have done this, Source Molecular is an older company that I think has gone out of business. You can't find them anymore. And now a company called Jonah Ventures. Years ago, Sally had mentioned when she was in there, they had done one test trial where they sent samples to Source Molecular and got results that didn't seem to make much sense. Just last year, um, Annapolis City did some of the funding. We did as the Environmental Center, and Arundel Rivers Federation did some funding to try to send some samples to get some answers. With the idea being that, well, this would be great, right? The pros, these are companies that have certifications and experience. They have, in theory, I'm going to say in theory several times now because did not work out the way we wanted it to. They have a large panel of targets. Like they have probes that we can say it's cow, dog, pig, even some specific birds, human, etc. And then it could potentially be quantitative, although they haven't moved to that. You collect water, you pass it through a filter, which should collect all of the DNA and some cells. You then extract it, break everything down, look for these targets, get numerical data that's going to give you an answer. So a couple of the cons, then I'll get to the results we had. It's very high cost. You have to send the sample blind. You don't know even sometimes what the concentration is. Like you assume it would be data that would be contamination from heavy rain, but if it's not contaminated, you sent it out and spent $300 on a sample and there wasn't much bacteria. It's a week. The data is going to be at least a week to find the source. Um, the issues that we've seen, it's not been developed enough to really track bacterial strains. It's much better for mammals and birds. So they do things like report data like, oh, we went out and we were looking at this river and we kind of looked at the number of people that were there versus the number of cattle. And when we took samples from water, that showed that we had a higher signature for people than we did cattle. But if we went to an area where there was more cattle than people, we had a higher signature for cattle than people, or yes, for cattle than people. The issue is we're not looking for cattle or human signals. We're looking for the signals from the bacteria in the gut. So this has become problematic. So we sent some data, sent some samples. This is a lot of data. I'm going to give you the long and the short of it. These are the dates. We knew that there was that the numbers of bacteria were high, the Enterococcus. We knew it was contaminated. Other than one Eastport Yacht Club, that was our negative control because we knew it was going to be good. That's sort of a negative control. They then looked for signatures for all of these species. Enterococcus, you would expect to correlate with the number in red, not be a perfect match because again, those are targets, not bacteria. And then to find out what the source of it was, they have a number of targets. So we tested for goose. Nothing came up that there was any goose poop associated with this, which was questionable to us. Dog. First of all, they still owe us free tests because they ran out of sample and didn't do all of the targets for some of them, which was very upsetting. So we tested for deer, we tested for dog. And I sent a sample that was just full of dog waste. So it was a positive control for dog. And yes, that matched, right? If I gave him something that was totally loaded with fresh actual dog waste, that worked. And there were some human targets. Now, keep in mind, this target is not for human feces, it's human DNA. And so when we started getting reports back, they wanted to say that, well, if the numbers come up high for human targets, that means contamination comes from humans. Well, that's totally false, right? People are in the water. You shed DNA from your skin. That's just saying that people had contact with the water. That has nothing to do with if it's bacteria from human feces. So they said, well, we have this new target. This new target is for a virus that infects pepper plants. Turns out humans are one of the only species that eat peppers. So you would have, in a high proportion, humans would be passing this out with their feces because you have the bacteria and the bacteria are infected by this virus. None of that came up positive. There was not a single thing that came from this company that really made any sense other than the one that we sent that was loaded with dog feces. And the human one only made sense because there was a lot of human DNA, but nothing that really told us that there was human feces in there. So what this tells us is that this source typing is still really very removed from reality. Um, Another problem that we see is there can be very local differences in strains of bacteria. So consequently, sending it to a company in another state that's developed these specific targets for bacteria that are where they are may not match with targets for the bacteria that we carry in our GI tracts, or our dogs or our deer. So this sending out to a company is really still not very useful to get to this answer. So we're still working through trying to find ways to do this. Um, having said that, as Sally has talks about, and how we've talked about also, we have a very good like working relationship with the Department of Health and Department of Public Works and things. They report when they find out, they report when there are sewage overflows. The vast majority of times when there are problems, high counts, it is not human waste, it's not sewage overflow, it's the runoff from impervious surfaces, which is another reason that we don't believe many of these results because we know a lot of it comes from geese. We know a significant amount comes from dogs. Those should have popped in a number of cases as positives. So the last question I just want to deal with is, are things getting worse? Every time we have a week like we did last week, I start sweating, getting upset. Oh my gosh, things are getting terrible. But this is actually a more complicated question than it appears on the surface. Um, for example, going back to this is just last week, and so there's this moment of panic. We have things in the 20,000s. It's just awful. Then we go back and I have to look at long-term data. So this bar graph shows like 9 years of data. And when we look at the tidal sites on Spa Creek, everything other than the ones very far upstream. When we look at the two non-tidal up in the freshwater, when we look at now Back Creek, which we only have 5 years of data from, and we look at, in this case, we're looking at the log of the average concentrations over the season. And what we see is in general, if we take all of the data for a season, like 19-ish data points, that there does not seem to be an overall increasing trend. That, you know, we have rain days, we have non-rain days, and they tend to even each other out. And on non-rain days, especially, things are not getting worse. We've done some initial work looking at some sites that we've been sampling at for like 15, 20 years, well, 15 years, and looked at all the data and then took out the rain days. And when there's not rain days, when we look at all the data and we look at just not rain days, it doesn't seem to be changing. The one thing that we have to go back and do a very careful analysis of is how bad is it after rain? Are the numbers higher after rain? And this is a student project that's going on now that we're still kind of we're still working through. On a limited sense, on the Magathie, so this is data directly from Sally's Magathie River Index from just this year. And she breaks down on the Magathie all the way back for 12, 13 years, that when you look at, again, all the dates and the days without rain, right, that the geometric means, right, they ebb and flow, right? But it's not just getting continuously worse. The one thing that catches my eye is last year for all the dates, which would have all the rain dates, there is a significant bump. So is it that after rain, there's so much contamination that those values are increasing, that that is something to look at? Like, is there more contamination going in? Is it because there are so many more geese, which their populations just seem to explode more and more every year? Is it because of more impervious surface? Is it because of more pet waste? All of these things may be contributing. So this is something that we still want to get to. Um, this is also showing, this was from a slide from an old study looking at sites up along the Magothy River after rain. And again, it wasn't because there was a place where there was a sewer leakage. The numbers were just high all along this upstream headwaters creek for the Magothy. Um, and then when we look at Spa Creek, right, every time I think how bad it can be, South Cherry Grove, which is that upstream site, right, we've had numbers higher than what we had just last week. So there is the ebb and flow. We're still trying to get at what the pattern is after rain, and especially trying to understand things like happened last week. So what are we still interested in? Every time we learn something, there's just a huge number of questions. Is the water quality getting worse after a rain event? To really understand that, we can't just look at data um, from all the sites over multiple years. We really have to focus on some sites that we've been doing the same sites many, many years. So Spa Creek is a perfect example for that. Magothy River, there are some sites that have been done for many years. Some sites we test some years and then we don't test other years. So we're really going back and looking at sites that we've tested continuously over many years. There are also sites on the main stem of the Severn that we're doing some of that analysis with also. Looking to see, is the average of the numbers after rain at these same sites getting higher and higher? Do bacteria persist longer as nutrient levels increase? That's something that we've been pondering. We don't have a lot of numbers. So this is like not just going out once and saying it rained yesterday, the numbers are really high. Like, is the number down below the threshold after 2 days, this 48 hours? There's some indication that as there's more eutrophication, higher levels of nutrients, that's not only allowing bacteria to survive, but if there's enough nutrients, are they actually reproducing, which would mean they would persist even longer. This is yet another problem that might come from increased nutrient levels. What are the sources of contamination? Still working on ways to figure this out. This is much more, we think, a research question than a question for a large company. We've talked to someone that does metabarcoding at CERC, Smithsonian Environmental Research Center, about we've done a little trying to develop our own primer, our own targets for locally. That is a big project. So we've talked to someone that does this type of thing all the time to see if there's something we can do for that. Um, and a question that we've had, this persistent question, as I mentioned, this IDEX kit that you buy from a company has become more the norm for the way that testing is done. It uses a huge amount of plastic, which as environmentalists, we should be trying to use less plastic. This bothers me. There's no way to go back and quality control after you have the result. If they're one vendor, only, the cost is high. So this summer we have access to one of those units. So we're going to do some side-by-side comparison of our methodology and that methodology to maybe convince more people to do it the old school way that's still very good. But with that, I'm going to wrap up. As usual, more questions than answers in these types of talks. But thank you for sticking with this technical talk. There's just a lot of different methods. And if you have any questions, I'm happy to answer them, or you can reach out to me at my email address also.